Hello everyone, this is Medical Gateway, and in this part we're going to study regarding the blood typing and cross matching. Okay, so what exactly is blood typing and cross matching? Okay, now blood typing is basically finding the blood group of a person, finding that which antigen is present on the red blood cells. Either antigen A is present, either antigen B is present, or both are present or both are absent. This is known as blood typing or blood grouping. Then what is cross matching? Cross matching is basically that whenever some person has to donate blood to the recipient, the blood of the donor and the recipient is mixed and checked for the cross reaction. Okay, or to be precise, the RBCs of the donor are mixed with the antibodies of the recipient to check for the cross reaction. Okay, now, so let's first discuss blood grouping and blood typing and how actually it is done. Okay, it is done in a simpler way that we take blood, which is going to be which we want to know the blood group. We take that blood and then we just centrifuge it and remove red blood cells from that blood. Then we take a slide, and on that slide we just place 2 drops of blood. One drop is this and the other drop is this. And on that 2 drops of blood, with one drop of the blood, we mix that 1 drop of blood with anti-sera A or anti-A antibodies, and the other drop of blood is mixed with anti-sera B or anti-B antibodies. And then we'll wait, and we see that which drop of blood shows coagulation and which drop doesn't show the coagulation. If the drop which is mixed with anti-A antibody shows coagulation, then it means that A antigen is present in the blood. And if the drop does not show coagulation, then it means that antigen A was not present on the RBC. And if the drop of the blood shows coagulation with anti-sera B, then it means that B antigen is present on the red blood cells. And if it doesn't show any coagulation with the addition of anti-sera B or anti-B antibodies, then it means that B antigen was not present in the blood. Okay, so now there are 4 conditions with reference to the ABO blood group system. So first one is this, that the blood is showing coagulation with anti-sera A. It means A antigen is present, and it is not showing any coagulation with the addition of anti-sera B. So it means that B antigen is not present. A antigen is present, B antigen is absent. It means that the blood group is A. So this is blood group A. This condition is blood group A, the first 2 drops. Then these 2 drops. Now, the blood hasn't shown any coagulation with the addition of anti-sera A. It means A antigen is absent. And the blood has shown coagulation with anti-sera B, which means B antigen is present. A antigen is absent and B antigen is present. It means the blood group in this case is blood group B. And then we have another scenario where the blood shows coagulation with the addition of anti-sera A as well as the addition of anti-sera B. Now in this case, it means that A antigen is present as well as B antigen is also present. So the blood group of the person is AB. And then we have the 4th condition, in which the blood doesn't show any coagulation with the addition of anti-sera A as well as the anti-sera B. It means no antigen is present in this case, so the blood group is O. Coagulation with anti-A means blood group is A. Coagulation with anti-B shows blood group is B. Coagulation with both A and B shows the blood group is AB. And no coagulation with both the anti-A and B means the blood group is O. Okay, and same is the case with Rh positive and Rh negative. If the blood shows coagulation with Rh antibodies, it means the blood group is positive. And if the blood doesn't show any coagulation by the addition of Rh antibodies, it means there is no D antigen present. It means the blood group is negative. Okay? A separate slide or a third drop of blood can be used for checking the Rh positive and Rh negative. And on the third drop, which is not made here, on the third drop, we will basically add anti-D antibodies. And with the addition of anti-D antibodies, if the coagulation occurs, it means the blood group is positive. If the coagulation doesn't occur, it means the blood group is Rh negative. Okay? So this is the process. The first, the RBCs are separated from the plasma. And why they are separated from the plasma? Because basically the antigens are present on the RBCs. So we want to study them. So RBCs are separated from the plasma and then they are diluted. And after being diluted, RBCs are mixed with anti-A antibodies and anti-B antibodies, just like this. So anti-A antibodies and anti-B antibodies are mixed with our red blood cells. Now, if the blood shows coagulation with anti-A antibody, it means this is blood group A. If the blood shows coagulation with anti-B antibody, it means the blood group is B. If the blood shows coagulation with anti-A as well as anti-B antibody, then it means the blood group is AB. And if the blood doesn't show any coagulation, then it means the blood group is O. Okay, so this was the concept of blood typing or blood grouping. Okay, basically finding out the blood group of the blood. Okay, now what exactly is cross-matching? Now in cross-matching, the donors and the recipient's blood samples are mixed with each other to observe any coagulation. This is a pretty direct way. If there is a donor, if there is a recipient, we just mix those two bloods and just see if the coagulation occurs or not. Basically, it is not the mixing of the two bloods. It is the mixing of recipient's plasma with the donor's red cells. Because we always have to see that whether the donor's red cell react with the recipient's antibodies. And recipient's antibodies are present in the recipient's plasma. So we see the reaction between the donor's red cells and the recipient's plasma. So why we are not considering the donor's plasma? Because the donor's plasma just dilutes itself, okay? Because of the relatively lower quantity as compared to the recipient's blood quantity. Okay, so in cross-matching, basically we're mixing the recipient's plasma with the donor's red cell, and we will observe for any coagulation. If the coagulation occurs by any reason, then the transfusion is not done. If this mixing of donor red cell and the recipient's plasma does not lead to any coagulation, then you are good to go. The blood group is matching, the blood typing is matching, and cross-matching is also giving a good result. There's no coagulation. Then that specific blood can be transfused to the other person. Okay? So this is blood grouping and cross-matching. The whole concept of the blood of this chapter of blood grouping and then studying this blood grouping and cross-matching is that, clinically speaking, we just want to know which blood can be transfused to the other person. Okay, so first of all, we have discussed the groups of the blood groups, and now we have discussed the grouping and cross-matching. In the next part, we will discuss regarding the transfusion reactions. Okay, so this was blood grouping and cross-matching. See you in next part.